Hello and welcome to my poster. My name is Clayton, and today I'm going to discuss the role of immune cell infiltration in IVD or intervertebral disc healing after injury. Chronic back pain is a debilitating disease that becomes more prevalent with age. A major cause of chronic back pain is intervertebral disc degeneration. The IVD is an essential connective tissue of the spine consisting of 3 tissues as shown in the red box. This structure will accumulate damage over time, and this, coupled with the low regenerative capacity of the IVD, can lead to degeneration. There are currently no disease-modifying treatments. Therefore, this study aims to identify the cells and pathways critical for IVD healing so that they can be used as targets for regenerative therapies to treat IVD degeneration. But in order to stimulate regeneration of a tissue, we have to understand the natural progression of healing. Tissue healing is a process that progresses in 3 general stages: inflammation, proliferation, and remodeling, and is characterized by the infiltration of immune cells, the upregulation of angiogenic pathways, and scar formation, respectively. The timing and types of infiltrating cells and the important pathways critical for IVD healing is currently unknown. In this study, I will determine the timeline of IVD healing after injury and the important immune cells and angiogenic pathways that stimulate healing. To do this, we conducted a longitudinal analysis of IVD healing after injury by using a needle puncture model to injury mouse tail IVDs and using uninjured tail IVDs as controls, as shown in the diagram. We then isolated IVDs 1 to 21 days post-injury, or DPI, and used qPCR to monitor immune cell infiltration or angiogenic pathway upregulation. Then at 7 DPI, we performed single-cell RNA sequencing by pulling 15 discs per condition from 3 mice to determine the genomic differences between control and injured cells. For the longitudinal analysis, at each time point, we used 3 immune cell markers, CD11B, CD3G, and CD19, and 2 angiogenic markers, VEGF-A and PDGF-alpha, to determine the temporal regulation of immune cell infiltration and angiogenesis over the course of 21 days. A simplified view of these data show that the peak time of immune cell infiltration is 3 DPI, with T cells having the highest upregulation. T cells also return robustly at 19 to 21 DPI. The inflammation stage plateaus by 7 DPI via myeloid cell infiltration, and this coincides with the beginning of the proliferation stage based on PDGF-alpha expression. These data together establish the temporal regulation of the stages of IVD healing post-injury. Seven DPI is a shared time point between the inflammation and proliferation stages. So we conducted single-cell RNA sequencing at this time to determine the transcriptomic changes that are driving the shift from a pro-inflammatory to a proliferative microenvironment. We found that of the 11 cell clusters present in both control and injured discs, 5 clusters expressed over 94% of the differentially expressed genes, or DEGs. When I analyzed these DEGs for their biological processes implicated using gene ontology, I found the majority of the upregulated DEGs are VEGF and T cell specific, while the downregulated DEGs are ECM and pro-inflammatory specific. Therefore, in this study, we observed a novel role of T cells in healing, where they come very early and then return very late, and determine the 5 specific cell populations that drive the transcriptomic changes seen at 7 DPI. And the DEGs at 7 DPI showed an increase in VEGF and T cell related pathways specifically, which corroborates the qPCR longitudinal data. For future studies, I will knock out T cells and myeloid cell populations specifically to determine their roles in IBD healing. And the novelty of this study is that this is the first study to profile the important cell types and signaling pathways involved in IBD healing, and these factors can be modulated to increase the regenerative capacity of the IBD. I would like to thank my lab, my co-mentor, collaborators, and funding sources, and thank you for your time. Please email me for any questions.